All right. Okay, anyone confirm me online? Am I audible to all of you? Online people confirm me. Anyone quickly confirm, am I audible online? Okay, not a problem. Let's continue. Okay, everyone is there, that's all, huh? Okay, so today we from today onwards we'll be starting two very important units, that is unit 3 as well as unit 10. So good afternoon, good afternoon. Okay, if everyone recall my words, I told to all of you unit 3 and unit 10 are two important units. When it comes to the unit 3, you know, I can use the word, it is one of the toughest unit where you can expect most of the analytical questions in the exam, right? And when it comes to the unit 10, it is a very lengthy, okay, and experimental. Surprisingly, you know, in the ecology more numerical questions also will come, at least two numericals will appear surely in the ecology. All right, so now first we will start with the most important unit, that is the unit 3. So after you know, after the first class gets over, we will go through the unit 10. Okay, everyone please continue. All right. So unit 3, generally call it as the molecular biology. What exactly we have to read in this particular chapter of the molecular biology? Okay, I think you all people read in your bachelor's as well as the master's. What exactly we have to focus on this specific unit? Everyone everyone remember. Let's start with this particular molecular biology. In this particular unit, according to our syllabus, first we have to start with an important process, that is the process of replication. And whenever we are reading the process of replication, we have to read the replication in the prokaryotes also as well as eukaryotes also. Then after the replication, that is the synthesis of the mRNA, that is transcription, again we have to read both in the prokaryotes as well as the eukaryotic organism. And the last and final one, which we call it as the process of translation, again we have to read in the both the prokaryotes as well as eukaryotes. And then last and final one, very important one, the gene expression, where in case of prokaryotes we will read about operon, in the eukaryotes we will read a very higher level of the gene organization. So what you understand by this lesson, we have to read everything in both prokaryotes also as well as in the eukaryotes also. In the exam, it is not necessary that they have to ask only prokaryotes and eukaryotes individually. 90% of the cases they combine both, they compare both, and they will raise the questions, right? This is, we have to read it. Okay, now what are the various reference books for this? The groundbreaking book for this molecular biology is Watson, definitely, there's no doubt about it. But if you want to read, you can read Weaver, you can read Alberts, even you can read little bit about the White and White also. If you're really interested to read the reference book, you can just read the Watson, that is more than sufficient. And I'll tell you one thing, listen, out of 100% friends, 95% you have to just understand. If you have a subject knowledge and application knowledge, definitely you can answer any question appears in this exam. So rather than studying first focus on understanding, then whole concept will be super clear. So let's start with replication first. So before understanding the replication, every one of you focus on this particular diagram. Entire lesson depends on this particular diagram. Whatever it is written here, we are going to read everything from very basic to the advanced level. Now tell me, if I ask you a question, what is the ultimate function of the DNA? Synthesis of protein. And we generally use a word that DNA is a genetic material which carries the information from one generation to the another generation. At the same time, ultimate function of the DNA is what? Synthesizing the protein. Now my question is very clear. DNA having the ability or capability to synthesize the protein. Correct na? And we read multiple times, protein is everything. Whatever the function performed by a living cell, how many cells we are having? A typical human being having 37.2 trillion cells in the body. Functions performed by every living cell is under the control of protein. An example, there is a continuous supply of the oxygen in the body. Who will supply the oxygen? Hemoglobin. Hemoglobin is what? Protein in nature. Correct na? Can you imagine a body reactions without enzymes? No. What are enzymes chemically? Protein. Whenever an antigen enters in the body, body starts releasing antibodies. What are antibodies? Protein. In simple words, functions performed by living cell is under the control of protein, and that protein synthesized by DNA. Now the question is here, even though DNA having the great ability or capability to synthesize the protein, it never involved in the synthesis of protein directly. What it will do? First it synthesizes an intermediate molecule, RNA. Then RNA synthesizes the protein. What is the reason why DNA cannot involve directly? What is the reason? Anyone? The simple reason, DNA is a highly stable molecule. Suppose if it's lost its stability or integrity, we cannot repair it back most of the time. An example, here RNA is involved in the synthesis of protein. Just imagine RNA is lost, or RNA got mutated, or RNA got damaged. We have what? DNA. Then DNA immediately synthesize the RNA, then we can synthesize this protein. Suppose example, while synthesizing the protein, if the DNA get damaged, you can't synthesize the RNA also, and you can't synthesize the protein also. So to protect its stability, to protect its integrity, DNA, instead of synthesizing the protein directly, it is going to synthesize an intermediate molecule which is called as RNA. Correct na? Now, understand very clearly, no confusions. So, before synthesizing the RNA, first DNA undergo a mechanism called as replication. We all look like our mother and father because their DNA passes from their generation to our generation. Correct na? So, DNA replication, what does it mean exactly? Duplicating the DNA. Now, once we duplicate the DNA, then from the DNA we are synthesizing RNA. See, the goal of the DNA is not at all to synthesize RNA, to synthesize protein. But in order to synthesize the protein, first it has to synthesize RNA. That's why DNA is going to synthesize the RNA. Now the question is here, everyone answer. Continue everyone. In order to synthesize the RNA, do you think we require whole DNA? An example, this is a DNA. Now all of you tell me, first this DNA has to undergo the process of replication. If the DNA wants to undergo the process of replication, do you think all the DNA is required? Remember, see, after the DNA replication, we get a daughter strand, correct na? So which means if the DNA wants to be duplicated, we require complete DNA. You're getting? See, this is the double-stranded DNA. It is getting open, or it is getting acting as a template strand. Now there is a synthesis of what? Daughter strand. Now what is the point? During the time of the DNA replication, whole DNA is involved. That is only getting duplicated into the daughter DNA. Now the question here, whenever we are synthesizing the RNA, there is no requirement of whole DNA. See, suppose friends, this is a DNA. DNA contain what? Many genes, group of genes. Now every gene can code for protein. Now from the DNA I have to synthesize the RNA. So I do not require whole DNA. I require a small piece or a small segment of the DNA. Which segment I require? DNA is so big DNA. So which segment I required? The segment which can code for a specific protein. An example, I want to code for hemoglobin. Then suppose example, this is the only part of the gene that can code for hemoglobin. Correct na? Only this small segment or a small part of the DNA only converted into RNA. You're getting? Then that RNA give rise to protein. Because this RNA is bringing the information only that contains for hemoglobin. Suppose example, I want to code for an antibody. So an example, this is the small segment of the DNA that can code for antibody. So only this portion converted into RNA. So depending on what kind of a protein is required, that specific gene only getting converted into RNA. The synthesis of RNA from the DNA we call transcription. In very simple words, we can say DNA mediated, or we can use the word DNA guided, or mediated we can use the word. DNA mediated RNA synthesis we called transcription. Now finally we got the RNA. So what is RNA is about? RNA is bringing the information from where? DNA. What information it is bringing? It is bringing that information that can give rise to protein. That's why we use the word messenger RNA. It is bringing a message that requires for the synthesis of protein. So now the most important here, this mRNA is involved in the synthesis of protein. It contains what? Information. That information is recognized by some amino acids. Because if I want to synthesize the protein, what I required? Amino acid. So that specific amino acid recognizes this mRNA. By using this mRNA, they synthesizes protein. We'll see that. So synthesis of a protein, in other words, RNA mediated protein synthesis, we call it as what? Translation. Can I also say this word? DNA guided RNA mediated protein synthesis, also called as translation. Because every information is coming from DNA. So everything is under the control of DNA. Correct na? But to synthesize the protein, we do not require DNA directly. What we require directly? RNA. Indirectly we require DNA. That's why it is called DNA guided and RNA mediated protein synthesis. Now don't confuse, everyone just try to understand. Whomever is listening very first time, undoubtedly it is too difficult unit. If you read once or twice, then it becomes so easy to all of you. Now once again understand. Now first, before synthesizing the protein, DNA synthesizes RNA. Before that, DNA undergoing the process of replication. Now the question is here, where this process of DNA replication occurs. I am asking about location in a different organisms. We are reading for prokaryotes and we are reading in eukaryotes. Tell me everyone, in case of a prokaryotic organism, where the DNA replication occur? Cytoplasm, because DNA do not have any well developed nucleus. Please come. And in case of a eukaryotes, because what about us? We are well developed organisms, right? We are having a well developed nucleus, then replication definitely happens in the nucleus. Whereas in case of a transcription, where it happens in the prokaryotes? Cytoplasm, because it do not have anything except cytoplasm inside the cell. Then in case of eukaryotes, again it happens in the nucleus. Whereas in case of protein synthesis, which is also called translation, where it happens in the prokaryotes? Cytoplasm. Every single reaction takes place in the cytoplasm. In eukaryotes? Cytoplasm. Because whether it is a prokaryotic organism or the eukaryotic organism, the process of the protein synthesis exclusively happens only in the cytoplasm, because the enzymes, the RNA which are required for the synthesis of protein, they are exclusively present within cytoplasm only. Correct na? Now all of you answered. Look into the picture. Now in case of prokaryotes, replication is over, transcription is also over. Then what do we get? mRNA. Correct na? Same way, in case of a prokaryotes are also, replication is over, transcription is also over, we get mRNA. Now please look into this. Let's consider this is a eukaryotic cell and this is a prokaryotic cell. Don't get confused. And the DNA replication means there is a synthesis of the DNA. Immediately what is coming out? RNA comes out. This is called as what? Transcription. So replication happens in the cytoplasm, transcription happens in the cytoplasm only. Now the very interesting point in case of a prokaryotic organism is that the moment a little amount of the RNA comes out, immediately what happens? The ribosome will go and attaches and start the synthesis of protein. So what does it mean? Which means the process of transcription, the process of translation, we called coupled, which means both are happening simultaneously, or we can use the word at a time. But let's consider a typical eukaryotic cell. We are having a very well developed nucleus. So definitely the process of replication happens within the nucleus. Now what is coming out? mRNA. Correct na? Now the question is very clear. Whatever the mRNA that is synthesized within the eukaryotic organism, this mRNA is not completely developed. We use the word immature mRNA. Tell me, all of you, we read in the BSc level, how can we convert this immature mRNA into mature mRNA? By using certain modifications. What modifications? You already read it. We call 5' capping, 3' polyadenylation and splicing. Now question is very clear, and these are all the modifications are happening after the transcription. That's why we call them as post processes. Now all of you answer, in case of a eukaryotic organism, where is the site of post transcriptional process? Where is the site? Within the, see, this is cell membranes, cytosol, and this is nucleus. Replication happened within the nucleus. Transcription happened within the nucleus. Post transcriptional modifications happens within the nucleus. And that after that, that mRNA which undergone the processing, finally they entered into the cytosol. There it undergo the mechanism of translation. Now tell me, where is the site of the post-transcriptional processes in prokaryotes? There are no post-transcriptional modification because there is no requirement. Why? Because the process of transcription, translation happening at a time. Correct na? But here, without modification, mRNA never ever enter into the cytosol. Correct na? Here no requirement of modification. This is only RNA, small stretch of the RNA come out, immediately ribosomes will bound and start the synthesis of protein. But here it never ever happens. Correct na? So now finally protein got synthesized here also. Again friends, however we called an mRNA as a pre-mRNA or immature mRNA, whenever a protein synthesizes, we call it as, an example we read now, when insulin got synthesized, what do we call it? Proinsulin. Why do we call proinsulin? Because it is not mature. Any protein that is synthesized in the human body, bacterial body, or any organism body, it has to undergo modifications. So those modifications are called as post-translational modifications. Where it will happen in prokaryotes? Cytosol. In eukaryotes? Cytosol. So I'll summarize all the steps now. In case of prokaryotic organisms, replication, transcription, translation, post-translation all happens in the cytoplasm. This is clear. In case of eukaryotes, replication, transcription, post-transcription we also called as RNA processing. Okay, and there is one more step we will read, RNA editing, that also happen within the nucleus. Then translation and post-translation happens in the cytoplasm. Correct na? There is a great possibility exams like SET or JAM, definitely we get the question from the location. So it is idealized to understand where exactly these locations are. Now let's quickly understand the enzymes, the key enzymes which are involved. Now ultimately first, before synthesizing the RNA, we are replicating the DNA. Now what is the major or versatile enzyme involved in the DNA replication? DNA polymerase. There are so many enzymes are there, but the principal enzyme involved in the replication is DNA polymerase. But DNA polymerase is always getting the guidance or information exclusively from DNA itself. That's the reason we use a word DNA dependent DNA polymerase, getting information from DNA itself. Now next, DNA is going to synthesize RNA. Correct now? Which means what I am synthesizing? RNA. What is the enzyme involved in the synthesis of RNA? RNA polymerase. But it never ever get the information from, it get the information from DNA. That's why we called as DNA dependent RNA polymerase. Correct now? Now you read multiple times, all the organisms except a few viruses, all organisms having DNA as their genetic material. Correct now? Which means by using DNA only they can synthesize RNA. But there are viruses in which RNA is genetic material, not the DNA. Which means friends, that time RNA also undergo the replication. You're getting my point correctly? So that time there is a requirement of an enzyme. Correct na? What enzyme? RNA polymerase itself. Correct na? And the same viruses, by using RNA, they are going to synthesize DNA. In general, we used DNA and we synthesized. That's why we call transcription. In other words, DNA mediated RNA synthesis is transcription. But right now, I am synthesizing, I am a virus in my body, I will always synthesize DNA by using RNA. So, RNA mediated DNA synthesis, since it is opposite to the transcription, so called as reverse transcription. Now the question in the exam, now all of you understand very correctly. Now what is the major enzyme involved in the transcription? DNA RNA polymerase. Now my goal is I want to synthesize from, so my goal is to synthesize DNA, so definitely enzyme is polymerase. You're getting? So what polymerase? DNA no. What I want to synthesize? But I am getting information from, that's why it is called DNA polymerase, also called as reverse transcription. Okay, so instead of remembering reverse transcriptase, remember this word, RNA dependent DNA polymerase, which synthesizes DNA from RNA. Correct na? Don't get confused. Okay, so DNA polymerase, RNA polymerase, DNA dependent DNA polymerase, DNA dependent RNA polymerase, RNA dependent DNA polymerase. Only these enzymes are involved in the process of replication and transcription. So now first we have to read extreme core depth of replication. So that is prokaryotes, eukaryotes, then we will go to second step and then we will go to the final step. And we can't read the process of replication directly. First we have to understand what are the various rules involved in replication. Then what are the enzymes involved in the replication. And then we finally enter into the process. Then we'll read prokaryotes and eukaryotes. It's a very, very depth. Okay. Now, this molecular biology having another name. we call it as molecular basis of inheritance. Why we have given this name? What is inheritance? Passing the characteristics by using molecules such as DNA and RNA. That's why we call molecular basis of inheritance. Now, ultimately information is passing from where to where? DNA to RNA. By that there is a synthesis of protein. This is called as central dogma. Central dogma is the mechanism by which information starts moving from the DNA to the protein level. Okay, so now you know very well, in the year 1953, James Watson and Francis Crick, they proposed double helical structure of the DNA. Correct na? With the help of what technique? With the help of X-ray crystallography technique, which was taken by Rosalind Wilkin and James Franklin. Later the Nobel Prize was shared among 4 people, right? See, whenever they have discovered double helical structure of the DNA, their entire research was published in the article Nature. And look into this, these are the last lines that they have written. What they have written, look into that. It has not escaped our notice that specific pairing we have postulated immediately suggests a possible copying mechanism. What do you mean by that? See, once they understand the helical structure, they also understand all the time base pairing will occur. Correct na? Between adenine and thymine and guanine and cytosine. This base pairing given them an idea or given them an hint. Okay, given them an idea or given them an hint that we have to propose a model by which DNA undergo the process of replication. Don't get confused. By the mechanism of base pairing, they understand that we have to propose a mechanism through which DNA undergoes the replication. We'll see that what kind of mechanism that they have proposed. So first we have to start with the process of DNA replication. Okay, it's a very broad concept. Please be patient and understand. So tell me, how can you give the definition of a replication? Give me a biological definition. See, this is double-stranded parent DNA. During the time of a DNA replication, each parent strand is getting separated. Whenever each parent strand gets separated, each parent strand acting as a template. And there is a synthesis of daughter strand. That's all. This is the biological definition. So, a reaction in which daughter DNAs are synthesized using the parental DNA as the template. That's all. See, what is the ultimate goal of the replication? Ultimate goal of the replication is transferring the information from one generation to the another generation with a, this is most important point, with the highest fidelity or highest accuracy. So in a simple words, DNA replication means it is a mechanism of synthesis of what strands? Daughter strands, by using parent strands as templates. This is a simple biological definition. Please give me the same definition in the form of a chemical definition. Chemical. See, when I ask you a question, DNA is made up of, what DNA is made up of? Nucleotides. You know very well, I told you how many nucleotides it contains, countless. But sometimes we use the word billions and billions. I told you na, if you take the DNA and keep it on the floor, it will go from planet Earth to the planet Mars 600 times. That much long molecule DNA is. So it is made up of billions and billions of nucleotides. Now see this, friends. So when one nucleotide combine with another nucleotide and so on it happens, then they can form the daughter DNA. See, when I use the word I am synthesizing the DNA, which means I am adding one nucleotide with another nucleotide. That's all the meaning. So now we already discussed many number of times. In case of DNA we have deoxyribonucleotides. In RNA we have ribonucleotides. Can you see here DNMP, deoxyribonucleic monophosphate. N represents number to which another nucleotide is added. Always, all of you remember without fail, whenever one nucleotide is adding with another nucleotide every time. Remember, nucleotide always come and add in the form of triphosphate. It never come and add in the form of a monophosphate or diphosphate. Triphosphate form only it will come and add. So here what happened, friends? Out of 3 phosphates, 2 phosphates are released out and 1 phosphate got added. So we called N plus 1. And so on, billions and billions of nucleotides will be added. Now, let's focus from this particular slide now. Before understanding the enzymology or process of DNA replication, first we have to understand basic rules that are involved in the DNA replication. Now, every rule is important and from this rule we always use it to get the numerical questions, okay, or experimental based questions. Number 1 rule, semi-conservative, we're going to read it everything. It has to start at origin. Synthesis should be always in a specific 5' to 3'. The direction of replication may be either unidirectional or bidirectional. Semi-discontinuous, requirement of primers. The process has to happen with high fidelity or accuracy. Now, before that, friends, we already know it very well, and just now we understand that DNA is made up of nucleotides. How does a nucleotide look like? Nucleotide contain sugar, base and phosphate, together called as one nucleotide, one. And I believe everyone remember, histones are what charged, friends? Positive or negative? Histones are positively charged, because what kind of amino acid contain? Lysine, arginine, histidine, positively charged. DNA is negative because of the presence of phosphates. That's why histones can wrap around the DNA so easily. Correct na? And if you look into the individual subunit, this is what sugar generally? Pentose sugar. So it is having how many carbons? 5 carbons, and we know that very well. And we also read multiple times, the second carbon contain OH group also and H. If second carbon contain OH group, that sugar is ribose. If they do not contain oxygen, we call it as deoxyribose sugar. And very important, we have 3 phosphate groups, alpha, beta, gamma, or mono di triphosphate. Now we already read that we have 2 type of nitrogenous bases, purines and pyrimidine. We know very well purines are bicyclic rings like this, and pyrimidines are only monocyclic rings. Now, most important point, we read now all these 3 as individual components. Now we have to combine everything. Now then only we call it as the nucleotide. Now all of you answer, how sugar can combine with the base? I told you in the nucleic acids also, no. The first carbon of the sugar combined with what? Nitrogenous base. In other words, friends, the bond between sugar and base, call it as what bond? N-glycosidic bond. We use the word glycosidic, means whichever is connected to the sugar, it is glycosidic. Why we use the word N? Because in the base it will connect it to the nitrogen atom. That's why we call it N-glycosidic bond. Now, 5th carbon of the sugar connected to phosphate group. So please don't confuse. You may call it as ester, or widely you can call it as anhydride. This never be phosphodiester. It is not phosphodiester. Okay. So the bond that connect sugar to the base is N-glycosidic bond. The bond connect the sugar to the phosphate, which is anhydride bond. Then what is exactly phosphodiester bond? Let's consider this is the first nucleotide. This is the sugar, and this is nitrogenous base, and these are the phosphate groups. Again, similarly, we have one more sugar, a nucleotide contains sugar, nitrogenous base and phosphate groups. And in the second position, what group we are having? It is RNA. Suppose RNA. RNA OH group. RNA is ribose, no. DNA only hydrogen. Whether it is DNA or RNA, third carbon, what they contain, friends? OH group. Correct na? What is the definition of a carbohydrate? It is what? Polyhydroxy. Because carbohydrates contain many number of OH groups. Correct na? So now at the third position, what group is present, friends? OH group. Now tell me, phosphate groups are present at which position? Fifth position. Very correct. Now let's understand what is phosphodiester bond. The bond formed between OH group and phosphate group. The third prime hydroxyl group which contain oxygen. Oxygen having a lone pair of electrons, which is acting like a nucleophile. What is that? Nucleophile. So oxygen nucleophilically come and attack on one phosphate. How many phosphates are left out? Released in the form of PPA. Whenever they are releasing, it generates energy, and that energy can be used to join this OH and phosphate. This bond called as phosphodiester bond. So phosphodiester bond is that type of a bond which is present, which is formed between two nucleotides. Correct na? Now friends, let's consider this is only the first nucleotide. Consider this is only the last nucleotide. Now all of you people answer, first nucleotide at the fifth carbon, which groups are there? Phosphate group. Now all of you answer, is this groups are involved in the phosphodiester bond? No. Yes or no? They are not involved. And can I say that this is a free group? Correct na? And which carbon it is located? That's why this is called 5 prime. Consider this is only the last nucleotide. So at the third carbon, which group is containing? Is this OH group is combined or involved in any phosphodiester? No. And it is located third carbon and it is free. That's why we call 3 prime. So one strand of the DNA exists in 5 prime to 3 prime. Opposite will be 3 prime to the 5 prime, depending on the availability of the free groups, either phosphate group or OH group. So this is what a simple nucleotide and how does it look like. So whatever I explained is a small basic which you read in the 11th standard. So now everyone focus from now. The number 1 rule, it seems to be little confusing and little tough also to understand. Focus on the concept correctly. If you get a question 99% set or net or any other exam, mostly you get experimental only. Hence you require a brilliant understanding of this concept correctly. So let's start with semi-conservative. What is this semi-conservative and what are all these models first of all? Just now I discussed Watson and Crick, they understand the double helical structure exclusively based on what? Base pairing. Based on base pairing only they understand DNA is exhibiting in the helical structure. Whenever they are understanding the base pairing, they got an idea. What idea? That they have to propose a mechanism through which DNA is undergoing the replication. They also don't know by which model or by which way DNA is undergoing the replication. But they thought that since they strictly follow the base pairing, so based on this base pairing, they proposed a mechanism through which DNA undergoes the replication. And that mechanism named as semi-conservative. Tell me friends, how can we give the definition of the semi-conservative? I already told you. According to the semi-conservative model, this is a parent DNA. Parent DNA containing both strands are getting separated. Once they get separated, each strand acting like a template, and there will be a synthesis of the daughter strand. That's why it is called semi-conservative model. Parent daughter, parent daughter. Correct na? But 90% of the other scientists, given approval for this model, whereas some other scientists rejected this model. Why? Because there is no evidence, there is no proof to prove that DNA only follows this model while it is undergoing the replication. Because there is no proof to prove that. They opposed that semi-conservative model, they proposed their own models. One is conservative model and one is dispersive model. What is actually a conservative model? All of you look into it. According to the conservative model, this is a parent DNA. Entire parent DNA getting preserved or conserved and is giving rise to daughter DNA. Here there is no separation of the parent strand. Entire parent DNA is going to give rise to daughter DNA. And there is a third model called as a dispersive model. According to this model, see here friends, we always use a word cut and paste model. See here, small daughter parent, daughter parent, daughter parent, daughter parent, like this. You can see here daughter parent, daughter parent. So this is dispersive model. So Watson and Crick, who proposed the double helical structure, they have given this semi-conservative, someone given conservative and dispersive. Now we have to understand, while undergoing the process of the DNA replication, originally DNA follows which model? While undergoing the replication. To prove this, there was a classical experiment was performed by Messelson and Stahl. Till today, this is one of the most classical experiment that happened in the field of biology or life sciences. Okay. Now, what is this experiment all about? Now, you only tell me, what is this experiment all about? What they have to prove? They have to prove that DNA, while undergoing the replication, it only follows what model? Semi-conservative model only they will follow. So how they have proved? Now first of all, what is semi-conservative model? Quickly recap. According to the semi-conservative model, parent strand is getting separated and giving rise to daughter strand. Every time remember, parent strands are always heavy. Correct na? Daughter strands are always light. You're getting my point. Parent strands are heavy, daughter strands are light. So now here combination of heavy also there and combination of light also there. So let's consider, this is a parent strand, both strands are heavy. Now during the time of a DNA replication, what happened friends? Both parent strands got separated and they have given rise to daughter strand, which means one heavy strand and one light strand, one more heavy strand and one more light strand. Can I use a word? Can I say this is an intermediate? Can I use a word? The combination of heavy and light, call it as intermediate. Correct na? I can also use a word hybrid. Correct na? Combination of both heavy and light. So our ultimate goal is only this. We have to prove this, that in case of a semi-conservative model, we have to form a hybrid DNA, combination of heavy and light. Now, how this Matthew Messelson and Stahl performed this experiment? First, they want to know which is heavy strand, which is light strand. I just explained it to you very clearly that a parent is heavy, daughter is light. I just explained it to you. Please come, sir, please. One is heavy and one is light. But how can we prove that? Okay, so in order to prove it, they have performed a very small experiment. Just a minute. Yeah. So now everyone just try to understand what kind of a good. What kind of experiment they have done. All of you focus. First, they have taken a test tube. In the first test tube, the test tube which contain a E. coli culture. E. coli is the bacteria, correct na? And this bacteria they kept under a medium of N15. What is N15? Heavy isotope, correct na? So we have a N14 and we have N15. N14 is a normal isotope, N15 is a heavy isotope, correct na? So first they have taken a test tube. In the test tube they have kept E. coli and that test tube kept under N15, which is a heavy isotope of nitrogen. And from here, what they have done? They have taken out a DNA. Correct na? And whenever they started observing the base, they found that it is strictly heavy. Currently understand, it is strictly heavy. But what is our semi-conservative model? We should contain both heavy and light. But when they performed, first time when they observed, we got only heavy. Now next time what they have done, again they have taken a test tube and again this test tube also contain abundant amount of the E. coli, but this test tube they kept under N14. And from this E. coli also they have isolated the DNA. and again they start observing the base. Then what they found, it is strictly light. Correct na? But our goal is to get heavy and light together, which is the hybrid. Later what they have done, later they have taken a test tube. This time what they have done, friends, they have added equal portions of N15 as well as N14. And to this they have added cesium chloride. Now question is very clear, what is cesium chloride? It is a salt. Now why we have added only cesium chloride, why not any other salt? The reason behind that, the density of the DNA, the density of the cesium chloride is almost similar, which is 1.91 gram per centimeter cube. That's why we have taken. Now tell me, in this test tube what are all present? N15, N14 and cesium chloride. Now what they have done, friends, they started the mechanism of centrifugation. What is centrifugation? Rotating at a higher speed. They have started rotating by using centrifuge machine up to 20 hours continuously. Now you tell me how the particles can be settled based on what? Density. Very good. So first which is getting settled? Cesium chloride. After that, N15 or N14? Then? Now can I conclude N15 is definitely heavy because it is settled before the N14. So compared to the N15, N14 is lighter. Here we can give a conclusion, parent strand is always heavy, daughter strand is always light. Based on what we have given this conclusion? Just based on the density. Based on what? Density. But our goal is not to separate heavy and light. Our goal is to prove how this heavy and light are forming together. In other words, how does this intermediate complex can form? So in a very simple words, to prove the semi-conservative model, the first step they have done is, they have to know which is heavy and which is light. To understand this, they have run the centrifugation. Then based on the density gradient, they proved that N15 is always heavy and N14 is always lighter. So with this simple knowledge, they started doing experiment and they started checking all the models of semi-conservative, conservative and dispersive. Now don't confuse, all of you focus on the picture. First, this is a beaker or a test tube, focus friends, which contain only N15, only N15, correct? Only N15. Now tell me Manoj, now it contain parent DNA or daughter DNA or combined? Only parent DNA, correct na? Can I use the word 100% heavy? Can I also say the band will be present on the top? No, where the band will be present? Down. Correct na? Band will be found always down because it is strictly heavy. So this is the first one. Now I want to prove 3 models, or I want to check 3 models. In order to check 3 models, what I have done, some of the N15 I have transferred to N14. Now this is the combination of N15 also, N14 also, correct na? Now from here, every 20 minutes I am taking the sample and I am checking what I am getting, whether I am getting only heavy, only light or only intermediate. Why only 20 minutes? Because I am checking it in the E. coli. What is the generation time of E. coli? 20 minutes. Correct na? This is called generation 0. In the generation 0, what we got, friends? All which DNA? Heavy DNA or original DNA. Now what happened here? Tell me now, you tell me. I got a band which is settled in the middle. What does it mean? See, if it is a heavy band or heavy DNA, where it is found? Bottom. We didn't get that. If it is light, top. So neither heavy nor light we got. What do we got is? Intermediate. Correct na? So which means which model? Which is semi-conservative model. Correct na? Now again after 20 minutes I did the same. Now again I have taken the sample. Can you tell me what we got now? Look into this. Both, what do we got? Light and wonderful. Can I say 50% light, 50% intermediate? Then next, same thing is repeating almost. Correct na? Or we can say more more light and intermediate. But can I give one statement? From generation 1, every time what is getting repeated? Intermediate is getting repeated. Which means semi-conservative model only it is getting repeated. Correct na? Now we have to disapprove the conservative model and dispersive. What is conservative? Entire parent DNA preserved or conserved and converting into? Did we get anywhere of that? Means entire parent DNA convert into daughter. And last and final one, dispersive. Small parent, daughter. Do we got anywhere? I mean, it's a combination of parent, daughter. We didn't get anywhere. So, with this simple experiment, what we can prove? We can prove semi-conservative model is the idealized model for the DNA replication. So, first is 0, which is generation 0, it is heavy. Generation 1, 100% intermediate. Here 50% light, and here what you can see 75% is light, 25% intermediate, and it's kept on repeating same. We no need to do that many generations also. 2 generations are enough to prove semi-conservative model only the correct model for the DNA replication. Okay, now everyone understand very correctly, there is no confusion. Slowly think and answer the given question. Slowly. It's one word answer. Make it simple. Sandhya, you did very well. Okay. Yes, Neelima, you try to see the questions once again. You answered questions whichever you know also wrong. Good. Well, Vagdevi also did well. But don't answer all the questions, answer only selected questions. Mamata, you also. Where is that girl? Mamata? She is not there. Done. Okay. Don't answer too many since it's a practice session, no problem, but choose it very carefully. You could have scored more. What's the answer? I told you multiple times, in order to answer any given question, make it simple. Don't think too much. What is the question? In the Michelson and Stahl experiment, what part of the DNA gets labeled with? What is that? Base. Correct, na? Nitrogen. See, just now in the previous experiments we have seen, no friends. This is a test tube. This is the E. coli culture. I kept under N15. Then from here, what I have isolated? DNA. And whether it is a heavy or light, how can I know? By measuring base only. That is only they are asking. Nitrogenous base. Correct, all correct. Slowly think. Forgot about answer. Tell me what the question is asking about. Where on the cesium chloride gradient would the following DNA is found? They are asking where these can be found in a medium. Positions you have to tell, that's all. First, DNA, double stranded DNA where both strands are labeled, which means it is very heavy. Heavy, lower. Where neither strand is labeled. Correct now, which is light. Light and high position. Only one strand is labeled. Position is middle, weight is intermediate. Correct now? They are asking only the positions. Slowly. 1. Why 0, man? Huh, 0. Rounds. Replication after switching to light n. Okay. Hence we have to consider this as the generation time. Generation time 0, 1, 2, 3. The time gap is 20 minutes. Tell me, generation 0, what do we got all? We got how many bands? Only one band, which is heavy. So it is settling at low. What do we got? How many bands we got? Intermediate means it is settled in the middle. How many bands we got? Only one. Intermediate doesn't mean that 2 bands. Where it is settled, that is important. Okay, only one band. Position? Second round. 50% of light, 50% of intermediate. We got 2 bands. Position? High, intermediate. Correct na? High and middle. Then can I say same? Correct na? Don't get confused here. None represents 0 generation time. Okay, or the starting interval. Okay, then 1, 2, 3 representing when N15 is transferred to the N14. Okay, so compulsory, whomever got the doubt, try to understand that very correctly. Okay, once again watch it. If you still find any difficulty, please ask me. Okay, a small question quickly. Others? What is the answer? If adenine is located on, this is adenine. Adenine pair with? Thymine pair with? Correct na? See, you have to consider in this way, adenine, thymine. Correct na? This is one base pair in one strand, in only one strand. Okay. Again, in other strand also there is a base pairing. Correct na? Thymine and adenine. Okay, do not get confused. You have to understand in this way. Then I asked this. On other strand is also this one. Correct na? If adenine is located on the strand Z, then on strand X at the same location must be. That's all. Adenine, thymine. Thymine original is going to base pair with adenine only. Don't confuse. Read one more time, it will become easy. Even though if you don't answer the question, no problem. Don't keep question mark faces. Look into it. Okay. Now, what is the number one rule is, what is the number one rule? Semi-conservative model. And the second and very, very important rule is, it should start at origin. Now, tell me, this is a very biggest DNA. Okay. And the process of the DNA replication never ever start randomly. It always starts at a point. The point at which DNA replication starts, we call it as ORC, origin of replication. And when we consider the organisms like a prokaryotes, they are very, very small. Hence, and what is the shape of the DNA in case of prokaryotes, which is circular, and the number of base pairs also very less compared to the eukaryotes. Hence, the number of origin of replications are only 1. Where in case of prokaryotes? In prokaryotes, there is only one single ORC. Whereas we human beings, let's consider higher eukaryotes, one origin of replication is not at all sufficient. to do the process of replication. We require multiple ORC. In generally friends, in case of a eukaryotes, instead of using the word ORC, we use the word replicator. Okay, instead of using the word ORC, origin of replication, instead of using that we use the word replicator, because we are having multiple sites, correct na? And whenever we are discussing the prokaryotic organism, always we take E. coli as the standard example. Similarly, whenever we are discussing about the eukaryotes, always we take yeast as the standard example. What is the reason? Yeast having 16 chromosomes, humans are having 23, almost, or some are less equal. That's the reason whenever we are reading eukaryotes, we will study yeast as the standard example. Now, in case of a yeast, this replicator having another name, we call it as ARS. Remember, don't forget, autonomously replicating sequence. Don't forget, replicator in every eukaryote, instead of ORC we call it replicator, whereas in case of a yeast, that replicator having another name, call it as ARS. That's all. That is the simple and simple second rule. And what is the third rule? Synthesis of the daughter strand should always happen in which direction? 5 prime to 3 prime. Can anyone give me the reason why? See, very, very good. What is the reason? Presence of 3 prime hydroxyl group. Very good. This is the 100% valid reason. Other reason? The DNA polymerase enzyme read the parent strand. I am using the word parent strand always in 3 prime to 5 prime. Always. Hence, daughter strand has to be synthesized in 5 prime to 3 prime. All of you please focus on the picture, because n number of times the question will appear from this, especially with respect to the experiment. Now, what is this friends? This is one nucleotide, this is the second nucleotide considered, and this is the sugar, this is the base, this is the phosphate, sugar, base, phosphate. And what is this group? OH group, oxygen having what? Lone pair of electron, which is called as nucleophile. Now, oxygen nucleophilically come and cut the phosphate group present at the 5 prime phosphate. Then one phosphate is cut, 2 phosphates are released out. When they are released out, they generate energy. And that energy can be used to join these two. This is what bond? Phosphodiester bond. So, in order to form the phosphodiester bond, the most important is oxygen or the OH group. Now, this is one of the reasons. Now, let's understand how actually DNA replication of a daughter strand happens in 5 prime to 3 prime, and we have to disapprove compulsory why it cannot happen in 3 prime to 5 prime. Now, look into the picture. This is a 5 prime and 3 prime. Tell me what group is present here? OH group. Very good. And in the starting of the lesson, I have told to all of you very clearly that whenever a nucleotide comes and added, always comes with how many phosphates? 3 phosphate. Always remember, nucleotides will come in the form of DNTP. Hence 3 phosphates are coming. Now this will come and attaches here. Tell me what is going to happen? This oxygen will cut 1 phosphate. There is a release of 2 phosphate and successfully added. Correct na? Can I say here OH group is there? Correct na? Yes. Now, just now it is very clear, daughter strand synthesis is happening in 5 prime to 3 prime, but proofreading, that is checking the error. In other words, whatever the nucleotide just added, that is the correct nucleotide or not, becomes to know by doing backtrack mechanism. I am doing the synthesis in this way. So, DNA is going out like this, but I am checking from backside here, because here only I added the nucleotide. So, I started checking here. During the time of a proofreading, it was understood that whatever the nucleotide we recently added, that is a wrong nucleotide. Then I will remove this wrong nucleotide. Then I have to replace with the healthy nucleotide. Correct na? Can I replace it or not? See, still here what group is present? OH group is present. Correct na? And this is the healthy nucleotide. Then what happen again? There is a cleavage of 1 phosphate 2 phosphates released in the form of PPI, and there is a addition of nucleotide. It is successful, no problem. But now let's see why DNA replication cannot happen in the opposite direction. Okay. Now this is hypothetical, never happens in the human body. Okay. It is all hypothetical, you have to understand. 3 prime to 5 prime. Now all of you answer. Can I say one line here? Here generally it should contain 3 phosphates. Correct na? Because 5 prime triphosphate. Now first nucleotide. This is the first nucleotide. First nucleotide also contain how many phosphates? And here what group is present? Wonderful, wonderful. It contains OH group. Very good. Now this is all hypothetical, never happens. And this OH group which is present in the nucleotide, it will go and cut 1 phosphate. Correct na? Then how many are releasing out? 2 phosphates are released out. Correct na? Only from here I am talking about. But the nucleotide having 3 phosphates. So it is successfully added. Correct na? Now again I am doing the proofreading. This side I am doing now because it is backtrack. Now during the time of a proofreading I have removed this. Correct na? Now I want to add one more nucleotide here. Correct na? Now can I add the nucleotide here? Is it possible? Tell me why. Very good. It should contain how many phosphates? 3 phosphates it should contain. Then only this OH group can go and cut this. Then only it can incorporate over there. Correct na? So what we can understand here, reaction does not proceed as no high energy bond would be cleaved. Correct or not? First nucleotide you can add successfully. During the time of proofreading you have removed it, which means already phosphate groups are also lost. You removed old nucleotide also. Now you are going to replace the another nucleotide. But in order to replace compulsory you require 3 phosphate groups which are already removed out. Hence new nucleotide cannot be added. It never actually happens in the body. All are hypothetical. The major reason for the formation of the phosphodiester bond is always 3 prime hydroxyl group. Then only phosphodiester bond can form. And what is the 4th rule? 4th rule. Good. So the process of the DNA replication is either unidirectional or bidirectional. This is also very, very important where we can, in the next class I will show you the questions where we get diagrammatic based questions definitely. Replication starts from unwinding the double-stranded DNA at a particular point called as origin. Now please look into this point. In case of prokaryotes and eukaryotes, mostly we follow bidirectional replication fork. An example friends, this is the DNA, this is the origin. Okay, and can you observe this highlighted area where the fork is moving only in one direction? That's why it is called a unidirectional fork. Suppose let's consider this is the origin. If you can observe the highlighted areas, the fork is moving in both direction, which is called bidirectional replication fork, which you people can clearly observe in the eukaryotic organism. Correct na? See here, this is one parent strand in 3 prime to 5 prime, another strand in the 5 prime to 3 prime. Correct na? So this is one daughter strand, this is another daughter strand, which is moving completely opposite directions. So this will be the one replication fork and this will be the another replication fork, which is called bidirectional replication fork. See, especially in case of a eukaryotic organism, whenever 2 forks are moving at a time, it look like a sleeping Y. A sleeping Y. This is what we can call replication fork. In other words, replication fork is the area or the point where active replication takes place. That is called as replication fork. Okay, in the process you will get it. We have to interconnect a whole lesson, then only the lesson will be very clear. And again, in case of a rules, the highest number of times the question appeared from the semi-conservative model. Later, highest number of questions appear from semi-discontinuous replication. Okay, what is semi-discontinuous replication? First of all, just now we discussed one thing very clearly. DNA polymerase enzyme read the parent strand only in 3 prime to 5 prime. This is very clear. According to the rules of the DNA replication, daughter strand has to be synthesized compulsory in the 5 prime to 3 prime only. Now all the students focus on the picture correctly. This is one parent strand, first parent strand, which is in which direction? 3 prime to 5 prime. Is there any problem for the DNA polymerase enzyme? No problem. Happily it reads the parent strand. And daughter strand is getting synthesized in which direction? 5 prime to 3 prime. That is also no problem, because the synthesis of daughter strand is happening according to the rules of DNA replication. No problem here. Okay. And can you observe the picture? The daughter strand is moving towards which direction? That is important. Towards the replication fork. Friends, this is the direction of replication fork. First of all, if you ask me, sir, how can we decide what is called as a replication fork direction? See friends, an example, this is the double-stranded DNA. Double-stranded DNA contain lot of base pairs, so they combine with each other. Correct now? Adenine, thymine, guanine, cytosine. Who will break the hydrogen bonds? Who will break the helicase? So an example, helicase is moving like this and breaking all the bonds. So this direction is called replication fork. The direction at which helicase is moving, back of the helicase only topoisomerase will move, back of the topoisomerase polymerase will move, and the replication will happen. So the daughter strand is moving towards the replication fork, correct or not? Helicase is also moving like this. Since it is moving towards the replication fork, it is moving continuously in a right direction. Whichever moves in the right direction we called leading strand. Since the synthesis is very much continuous, we call continuous synthesis. Now tell me, other parent strand is in which direction? Firstly, parent. First parent strand is in 3 prime to 5 prime, no problem. Next parent strand is in which direction? There is a problem here, because DNA polymerase will read only in 3 prime to 5. And most importantly, daughter strand has to synthesize definitely in the 5 prime to 3 prime only. Correct na? Hence, replication fork is in this direction. But daughter DNA is synthesizing opposite to the replication fork. Correct na? So that's the reason we call it as lagging strand. And we use the word discontinuous synthesis. Correct na? So why we use semi-discontinuous? Half is continuous, half is discontinuous. That's why we use the word semi-discontinuous. We'll see in the process. These are all just rules, that's all. We haven't studied any process. So the interesting point here, whenever semi-discontinuous synthesis is happening, instead of a continuous manner, they are always synthesized in the form of a pieces. Means, what is the meaning of a pieces? This is a small primer and small nucleotides. Again primer, again nucleotides. So they are always synthesized in the form of a pieces. These pieces only called as Okazaki fragments based on the name of the discoverer. I will explain crystal clearly in the process how actually they got formed. So all of you please looking into this. Leading strand synthesis is continuous, correct na? This is one parent strand of 3 prime to 5 prime. Definitely daughter strand is in 5 prime to 3 prime. And can you see this picture, friends? This is the direction of replication fork. So since the strand is moving exactly at that direction, we call continuous synthesis and leading strand. Another parent strand is located in 5 prime to 3 prime. But originally, according to our rule, daughter strand has to be definitely synthesized in, but it happens in a opposite direction. Means this is the direction of replication fork, but it is happening opposite. Away from the replication fork, that's why we call it as discontinuous. Instead of happening continuously, it happens in the form of a pieces. We called Okazaki fragments. Now please don't confuse. In case of a prokaryotic organism, what is called Okazaki fragment means? It is nothing but group of nucleotides only. Based on the name of the discoverer, we used the word Okazaki. One Okazaki fragment is equal to 1000 to 2000 bases. In case of prokaryotes, total how many Okazaki fragments are present in the prokaryotes? Very less. We can use the word few thousands. Let's consider 10,000. 10,000 Okazaki fragments are present in the prokaryotes. Each fragment is 1000 to 2000 bases. But when I consider the eukaryotes, friends, one Okazaki fragment is how much? Very small, 1000 to 200 bases, but they are present in millions in number. Okay, so lengthwise prokaryotic Okazaki fragment is high, number-wise low. Here it is lengthwise small, number-wise millions in number. Please don't confuse. Okay, next, the most important rule here also. Okay, RNA primers are required. What is the ultimate goal of the DNA polymerase? Synthesis of daughter strand. What is the meaning of synthesis of daughter strand? I said adding of nucleotide, addition of one nucleotide to the another nucleotide, that is only we call it as synthesis of daughter strand. In the olden days, we might observed many times in the villages especially, we have a borewell pumps. Remember, what is the speciality of a borewell pump? First we will keep little amount of water in the borewell pump. Upon applying the suction or pressure, we get continuous. Correct na? Same way polymerase also will work. Ultimate goal of the polymerase enzyme is what? Adding one nucleotide with another nucleotide. So someone has to add few nucleotides. Then it will take another nucleotide and keep on adding to it. So you can see here, this is one parent strand. See here, we have added a small group of nucleotides. Correct na? We call primer. Primer nothing but, friends, a stretch or a small group of nucleotide. Let's consider a eukaryotic organism like a human being, or consider prokaryotic organism only. Hardly we will add 11 plus or minus 1. Hardly 12 nucleotide bases we added, that's all. Then immediately DNA polymerase will come and continues this. You are getting? No need to get confused. So now my question is that what is primer? A small stretch of nucleotide, and that can be continued by the DNA polymerase. And from where the nucleotides are coming from? Nucleotides are there inside the cell. Cytoplasm contains nucleotide, nucleus contains nucleotides. Nucleotides nothing but what? Sugar base phosphate. They are already available. So it will just take it and keep on adding. Now my question is, why we have used the word RNA primers? Because we are synthesizing DNA, but we are adding the primer of? The reason we do not, there is an enzyme called as RNA primase. RNA primase is the enzyme which add RNA primer. Whether is it a prokaryotic or eukaryotes, we don't have any enzyme that is existing named as DNA primase. There is no enzyme. That's why first we add the RNA primer. Then the question is that, sir, what is the use of RNA primer in the DNA? Of course there is no use. Once the DNA replication is over, we'll remove it out. That we will read in the process. To start the process we added. While ending the process, definitely we will remove it. Now, what is the difference between the leading and lagging strand here? In the leading strand, friends, how many fragments we have added? One primer I have added, and I have continued with the help of DNA polymerase. But in the lagging strand, multiple primer. Can you see the picture? The black color one is the primer. And after that, what fragment I added? See, primer is also nothing but nucleotides only, but there are RNA nucleotides. Later we added DNA nucleotides, which is Okazaki fragment. Again I will add primer. Then again I will continue Okazaki fragment. Primer, Okazaki fragment. At the end we will remove all the primers. Then there is a gap. We will replace with DNA nucleotides. That we will read in the process. So primers are very important for the process of DNA replication. Then the last and final simple rule is replication fidelity or accuracy. Very important this one also, because whatever the we read in the genetics, even though one nucleotide or one base got damaged in the coding region, it leads to non-functional protein or hyperactive protein. Hence, entire information of the DNA has to be replicated with the highest accuracy. So to maintain that highest accuracy, we follow two mechanisms. One is proofreading and base selection. Anyway, base selection we know that this one, if this base selection is not happening, or the DNA is not following this base selection, will stop the replication immediately. And what is the another mechanism we read? Proofreading. Just now we discussed that DNA synthesis is happening in which direction? 5 prime to 3 prime. But proofreading happens at 3 prime to 5 prime. So while doing the proofreading, if the DNA polymerase enzyme identifies that added nucleotide is the wrong nucleotide, it will stop the replication, remove the wrong nucleotide, replace with the healthy nucleotide, and then only replication will be continued. So by using proofreading and base selection, we follow the accuracy or fidelity of the DNA replication. Whatever we explained so far is a common basic rules, but without understanding the rules, you can't answer the given questions in the exam. Quickly answer. Oh, 249. Correct. Now reason, we have a 3 hydrogen bonds. Quickly answer. Sorry. First one. Others? Everyone. Okay. Look into the given question. A segment of the lagging strand of the DNA molecule has this sequence. Okay. So let's write it. Consider this is leading strand. This is lagging. Correct, na? So let's write that sequence. Tell me. 3 prime. Correct, na? This is the first statement. Next, so this is what strand now? Leading. So parent strand should be in? This parent strand is in? Correct, na? It is clear. Now this line is clear. Now read the next line. According to this, what should be the sequence of the DNA that is for the? So now for this leading strand is in which direction? Can I say opposite sequence will be there? Correct or not? Can I say 5 prime to 3 prime and all are opposite? Correct? Huh? And what is the question? They are asking the template strand of this leading strand. This is leading. What is the template for this? This one. Can I say the sequence will be same? Correct or not? What will be the answer? Correct, na? What is the question? Once again, understand everyone. They have given a sequence of what strand? Lagging strand. Correct, na? Okay, I'll draw quickly. This is one parent strand, and this is what strand? Leading strand. Then this is what strand? Another parent strand, and this is lagging strand. Correct, na? Now what sequence they have given? They have given this lagging strand sequence. They are asking the sequence of what? Parent strand, template of the leading strand. Correct, na? They are not at all asking the sequence of the leading strand. They are asking the sequence of template of the. Suppose example, this is the sequence. In the leading strand, can I say opposite sequence? Correct, na? Then again for that template, what sequence will get? Will get the same. You are getting the point correctly or not? So obviously answer will be the C. Correct, everyone is correct. Correct, na? So don't get confused. Okay. So these are all what? Small basic rules of the replication. Okay. In the next class, we'll start with the most important one, enzymes involved in the replication. Okay, everyone take exactly 10 minutes break. We'll start it immediately. Okay, everyone join at 3:10. I'll continue the ecology. Okay. 3:10. Okay, through the another YouTube link whichever I have provided. Okay, please join sharp 3:10.